Hello, thanks for joining me for this TouchCast. The purpose of this video is actually to recap or to summarize hopefully some of the main ideas that you took away from completing that cell differentiation and specialization POGL activity. I know we've been learning about mitosis. Now some of these ideas that you were just exploring and investigating can get a little bit complicated. So I just wanted to make sure that things were clarified for you and that they were making sense, all right? So let's begin. So multicellular organisms are made up of many cells, right? But amazingly, they begin as a single fertilized cell. That single fertilized cell is called the zygote. Now that single fertilized cell is going to divide over and over by mitosis to form embryos. And then eventually those embryos are going to develop into mature organisms that are made up of the many cells. Now this is true for all multicellular organisms. There's a model here showing you the animal development, and this one's for an adult sea star, right? And there's one on the bottom that just shows you plant development. Now, while their pathways might look a little bit differently, you can see that both of them begin with that zygote and divide over and over again until there's enough cells to form this complex organism. All right, we also learned that multicellular organisms are composed of different types of specialized cells. Humans, for example, have an estimated 200 plus different specialized cells. From last unit, we know that specialized cells can perform specialized tasks because they have special proteins, right? The proteins allow them to do this work, and different cells manufacture different proteins. Now, because specialized cells carry out specialized functions, they actually have specialized structures. And that's another theme that we've kind of talked about, right? Is that form follows function, or structure dictates function. In this model over here, we can see that we have an egg, an ovum, right? And it's being fertilized by the sperm. Now, what's going to be created? It's going to be that zygote, that single fertilized egg. And from that one single fertilized egg, we get all these multitude of different cell types, right? Bone and muscle and intestinal. You can recognize those as being kind of the villi right there, right? On top of those specialized epithelial cells. Fat cells, smooth muscle cells, red blood cells, nerve cells, loose connective tissue with fibroblasts. I mean, all of this from one single cell. That's pretty amazing. So the question is how is that possible? Where do all those specialized cell types come from? How does this happen? How do we get so many different specialized cells from one fertilized cell, one zygote, right? Well, specialized cells are actually formed through the process of differentiation of unspecialized cells. Those unspecialized cells are known as stem cells, right? And if you look at that word, that word differentiation, we can see another word in there, right? And that other word being different. And that's because this is the process of becoming different is all that means. So we have the stem cell, and we can see there's a couple different fates here. So that stem cell, that undifferentiated cell, could divide by mitosis and create two daughter cells. One of those daughter cells can remain as a stem cell, right? So you always have a constant supply of stem cells. Well, that other daughter cell could actually go through that process of differentiation and become specialized cells, right? Like we have a neuron there and a muscle cell. Now, one thing that you should be aware of is that in general, the potential of animal stem cells, like our human stem cells, to differentiate into many different types of cell types actually decreases as an organism develops. In other words, is that the older an organisms get, animals get, in usual, those cell types lose their ability to differentiate or become anything they want to. They can still, if they're stem cells, turn into some different types of cells, but not as many as they could when back earlier when the cell was developing. Now, same is not necessarily true for plant cells. Plant cells seem to retain their ability to differentiate into many different types of cells, but in animals, this tends to be kind of a general rule of thumb, although there are exceptions. Now, how are they able to differentiate? Well, you need to remember, this is very important, is that all cells contain the same DNA, right? All cells contain the same DNA. we know through the process of cell division, through the process of mitosis, the DNA is going to replicate. It's going to copy itself so that each daughter cell gets an exact copy of that genetic information. So if all cells have the exact same DNA, how are they able to have different structures and functions? Well, that's because cells can express different genes, right? And that's what this model is showing, and this is one from that pogal activity right here. So we have red blood cells, a muscle cell, and a pancreatic cell, right? And we can see that all of them are expressing this gene for housekeeping. Housekeeping just meaning to do the regular things like respiring and cleaning up the cell and just maintaining order, right? Keeping things alive. But we can also see that hemoglobin, that gene that makes hemoglobin, that particular protein, is only turned on. It's only expressed in a red blood cell, right? When we say expressed or differential expression, it just means the gene is turned on. It means that proteins, mRNA, is being created from that gene, all right? So again, red blood cells are creating hemoglobin, but we can see that muscle cells, that's not shaded, they are not creating that, and pancreatic cells, they're also not making hemoglobin, right? So they're not expressing that particular gene. If we look at a pancreatic cell, we can see that the gene for insulin is actually being expressed. And you remember from insulin is a very important protein that's involved in regulating blood glucose levels, right? We learned about that earlier in the year when we were discussing negative feedback mechanisms in homeostasis. And then you can see on muscle, myosin, this protein right here, this is created because the gene is expressed for myosin in muscle, but because the red blood cells and the pancreatic cells have a different function, they don't need to express that gene. Now, here's another model that's kind of showing the same thing. So we have our undifferentiated cell over here. Now remember, that's going to be our stem cell, right? So we have our undifferentiated cell, and then coming out right here, we have a representation of DNA. In this undifferentiated cell, we can see we have different colored regions of the DNA, the A, B, C, D, E, F. Well, those letters and those shaded regions all represent different genes. And if there's an X over those genes, that means the gene is not being expressed. That gene is kind of turned off. There's no transcription happening, there's no translation happening, right? If there's an arrow to the right, like we have right here, that means that gene is being expressed, that there's protein being created. So we can see in A, that particular gene is being expressed, and gene C over here, that gene's being expressed, and even to a greater degree than gene A. There's a lot of transcription and translation going on there. Well, these stem cells are going to be expressing these genes, and then they can go through the process of cell division, right? And remember that one of those daughter cells will stay as a stem cell, and the other one can actually differentiate. And through the process of differentiation, we can get muscle cells, nerve cells, and epithelial cells. Again, looking down here at the sections of genes in each of these different cells, we can see that in muscle cells are actually activating or expressing different genes than nerve and epithelial cells. Not only are different genes being expressed, that different genes are being expressed more. That is, there's more transcription and more translation of some genes over the others. And this is what allows cells to be specialized, to take on different roles, to look differently, even though they all have the same exact identical DNA. So I hope this video was helpful at pulling out and maybe just summarizing, helping to focus on the main ideas of that POGL activity. Now, I did provide for you an answer key, and I would really like you to go and compare your responses to the answer key that I provided, to not only look at the wording to make sure that you understood the questions and you had all the answers right, to evaluate your understanding. Now, if you do have any questions about what we just talked about, please come prepared to ask those during our next Zoom conference, or as always, you can post those on our community page if you haven't accessed that already. All right, so take care. If I don't see you, have a great weekend, and I hope to see you soon. Bye.